Okay, good morning, everybody. And we're going to carry on with our discussion about the protists this morning. We ended up more or less with this slide last time. And what I want to, I'll just remind you about some of the aspects of this rather strange grouping of the protists, or the proper name, the protista. It's a very, very diverse group, and we know that it's actually a poor grouping, that some of them simply don't belong together, but we're not sure where else to put them in the scheme of all living things on Earth. Let's have a look at the different groups that we're going to talk about. We're going to talk about the excavata, the chrome alveolata, the rhizaria, the archaeplastida, and the uniconta. And the very, very diverse. In fact, there is more diversity within the protista, really, than there is in all of the rest of the eukaryotes. So it's a very important group, and they're important in all sorts of ways. They're very important ecologically as well, as just in terms of their evolutionary significance and everything else. So I just want to point out a couple of things about these groupings. Have a look here at this group here, the excavata, and you'll see that this is a recognized grouping or clade. That is, they share a common ancestor. They share a common ancestor somewhere here. And we see that common ancestry actually reflected in some aspects of their morphology. So fine, there's a grouping there that we recognize as a distinct clade. Here is another one here, and here is another clade here, each with their common ancestor. But in this grouping here, the chrome alveolata, we believe that these two groups are closely related and shared a common ancestor. So this is itself, we believe, is a clade. Although I will say, you'll notice that these are dotted lines. We don't know what the ancestor was. We have no idea what but we assume that there was a common ancestor here. So where you see them in these neat groupings like this, that is a distinct clade. The assumption is they share a common ancestor. Have a look over here, and you'll realize, looking at whenever you see a phylogenetic tree, which is what this is, with this kind of linkage between groups, this is effectively telling you we don't know. We know nothing. We know nothing about the common ancestry of all of these groups together. We do not know anything about how this group here is related to this group here. We can say something, we can guess that this group here is related to this group here, but we cannot say how this whole grouping here is related to this group here. What ancestry they share or do not share, we simply don't know. And again, this whole grouping, the protista, these are eukaryotes, which are not animals, which are not plants, and which are not fungi. Please ignore, for the moment, just down here, you'll see there's fungi and animals in this group, the uniconta. Just for the moment, ignore that. That's why they're not highlighted. Only the groups which are highlighted in this diagram are actually protists. The others are not. We'll understand why these are here towards the end of this discussion. Okay, so let's have a look. We're going to go group by group. And so we're going to discuss the excavata first. And there are three groups here: diplomonads and parabasilids. These, we really just put them into a single group. They're closely related and very, very similar to one another. And I'll show you an example of each, but just put that's why I put this little bracket here. Anything with an asterisk next to it, that is something we're going to discuss. There are some which we will not discuss. Okay, so diplomonads, parabasilids, and euglenozoa, they all belong together in this group called the excavata. And we're not going to discuss what the morphological details are, but the excavata all have some strange aspects to all of their cytoskeleton, which is unique to the group. And none, despite the fact that they share that, and we believe they are related to one another, the exact relationship is not known. Okay, we don't know exactly how they related to one another or derived from one another. So let's have a look at a diplomonad. This is Giardia, and some of you may have had experience of Giardia. Giardia is very common, and it's a protist which occurs in water, in fresh water, especially. And when you drink it, it causes diarrhea, a violent diarrhea. You can develop an illness. It breeds in the gut. And you can see here, here's the cell here, and characteristically for this group, the diplomonads, it has two nuclei. There's one there, and there's one there, and it has numerous flagella coming off it there, here, and it forms this little smiley face, very easy to recognize under the microscope. And I'll tell you a little side story to this. This is one of the organisms which Van Leeuwenhoek, you remember Van Leeuwenhoek developed the first a really high quality microscope and examined water and all sorts of things. And Van Leeuwenhoek had a violent diarrhea, and he examined it under his microscope. And lo and behold, he saw and drew very, very accurately Giardia cells, which is a remarkable feat because they're actually very recognizable, but fairly difficult to find in a sample. So this is Giardia, very, very common, and it causes a disease. It's often called hiker's diarrhea because people out hiking get thirsty and they drink water from a natural water source. Giardia is commonly found there. It's not a serious illness, by the way. It's usually self-limiting, but it can be chronic. Nonetheless, a very common cause of it. Here it is here. You can see there's got a big sucker here. And with that sucker, it sticks onto this wall of the intestine. These are all the flagella. It has numerous flagella. So Giardia intestinalis, that's your typical diplomonad. Here's a closely related organism, but this is a parabacillid. And this is Trichomonas vaginalis, as its name implies. This is an organism which is commonly found in the vagina. It's resistant to the acids, the lactic acids, which normally preserve the normal flora of the vagina. And it is sexually transmitted. And Trichomonas will commonly, it's quite common, and it will cause a very purulent, smelly discharge with a tremendous itching. Its primary symptom is produces an unbearable itching. So it's Trichomonas, and that's a parabacillid. You can see how similar it is in structure. A single cell, lots of flagella, and closely related to Giardia. Okay, so the last group there in the excavata that we're going to talk about are the Euglenozoa. Now, this is a tremendously important group. Very, very big group, very, very diverse, as you'll see in a minute. There are all sorts of feeding mechanisms in the Euglenozoa. There are predatory heterotrophs, many photosynthetic autotrophs. That is the typical euglena cell, which some of you may have already seen. And then a whole group of strange parasites as well, which we'll see. There is, again, for this particular group, the Euglenozoa, there is a single feature which puts them into a group, despite the fact that they are so diverse, puts them into a single group, and that is that there is this strange structure inside at least one of their flagella. and it's a crystalline rod. I'll show you a cross-section in a second. So that tells us that these share that feature from a common ancestry. So here's a typical Euglena cell, just a single cell with prominent big flagella. Actually, most of them swim this way, like that, in that direction, not that way, which you might think was logical. Most of them swim in this direction here. And here's the flagellum cut through in cross-section, and all of this here, there, that's the strange crystalline rod whose function actually is not clear either. It was not really known what it does. But here's you see these little tiny tubules cut across here. This ring of microtubules, that's the actual mechanism whereby the flagellum beats. That's the flagellum, flagella mechanism itself there. So this is the typical Euglena cell, which you would see in lab, for example. Very, very common. You see many euglenoid cells, probably many different species that look pretty much the same, freshwater, in marine systems, but also just in damp soil, that sort of thing. You commonly see them. They may become so numerous in sands, for example, that they may form a green film on the sand. So here's the cell itself, a single cell, obviously with a cell membrane, and it has a cell kind of cell wall around it as well. Here's the flagellum here, there. The number of flagella varies. It varies by species, but it also varies within species. 1, 2, even more flagella can occur. So here's a very common and prominent feature, and it's this red dot here. And it's an eye spot. It's called the eye spot, but it doesn't really see an image. But it perceives light. And its position right next to the flagellum here is not accidental, because what this eye spot does is it governs the beating of this flagellum. It governs the direction in which the organism swims, and its responsibility is to keep the cell in an optimum light condition for photosynthesis, because this cell is packed with chloroplasts. You can see them all here. This is a photosynthetic organism. The Euglena itself is photosynthetic, but it's also heterotrophic, and it can open. You see this kind of passage here around the flagellum there. It can actually open that up like a mouth, like a gullet, and it can actually swallow things from the water. So it has a heterotrophic ability as well. And if you keep Euglena in the dark for long enough, provided you feed it with other things to feed on, it can eventually lose all of its chloroplasts and become totally heterotrophic. So it's quite a strange thing. It has a single nucleus there, many, many chloroplasts. So just remember these different features. The cell wall is not structured like just a single sheet like cell walls often are. The cell wall consists of bands of material that's wrapped around the cell, and that is called the pellicle, which is actually a part of the way that it resists osmotic pressures. It's not the only way that a freshwater Euglena would resist osmotic pressures. They also have this here. Oh, sorry, where am I? No, I'm sorry, it's not it. This here, which is a contractile vacuole. And the contractile vacuole is a pump. It's a water pump. That's all that it is. It continually absorbs water that is pouring in due to osmotic pressure, and then just voids it to the outside. you can actually see it pumping continuously to pump water out. So that's euglena, very typical, relatively simple cell. But here, believe it or not, is a relative. And this is a group called the kinetoplastids. There's all still in the euglenozoa, but it's a kinetoplastid. And this is trypanosoma. This is a blood parasite. It is a blood parasite which infects various vertebrates, especially antelope, cattle, that sort of thing, in Africa. And when transmitted to human beings, it causes trypanosomiasis, or sleeping sickness. And it is transmitted by the bite of a fly. So it's the kinetoplastids are just, first of all, you can pick, this here is the cell body, almost like a euglena, right there. But then it has a flagellum, which emerges here and then wraps itself all the way around here. Here's the flagellum there. And here it goes down here. And there's a membrane attached to the flagellum and it forms what's called the undulating membrane, and that's how it locomotes. Instead of the flagellum beating free to move it through the medium, the flagellum, when it beats here, it waves this membrane like this. And that's how it locomotes. And that locomotion is driven by a giant mitochondrion, which you can see here. This is a huge mitochondrion, specially modified mitochondrion called the kinetoplast. That's where it gets its name from, kinetoplastids, because it has a kinetoplast, a modified mitochondrion. A very, very dangerous disease, by the way. In Africa, it causes widespread illness and death, and also economic disruption because it infects cattle. So, its normal hosts are wild animals, wild antelope. Here's a beautiful scanning electron micrograph of trypanosoma. These are red blood corpuscles here in the background, and here are the trypanosomes themselves there. So that's trypanosomiasis. There are other trypanosomes, by the way. One for biologists, which is of particular interest, is one that occurs in South America. It doesn't cause sleeping sickness, but it causes a disease called Chagas disease, which is an chronic inflammatory disease, and it's transmitted by bed bugs. The reason that it's interesting to biologists is because many people believe that Charles Darwin acquired Chagas disease when he traveled to South America on the voyage of the Beagle. And that was one of the reasons when he arrived back in England, he was extremely depressed and stayed that way for many years and failed to finalize his work from the voyage until like 20 years later. Many people believe that he left as a vigorous, energetic young man, he came back as a depressed, fatigued, much older man. And many people believe it was because he had acquired a trypanosome disease in South America called Chagas disease. Okay, so that was our excavator, okay? Now we're going to move to a group called the chrome alveolata. Again, united by a strange feature of morphology, and this is that they possess a structure inside the cell membrane, which is called the alveolus. And so they're all alveolate. There are two distinct groups. In this group, the chrome alveolata, there are two distinct clades, one called the true alveolates and the other called the straminopiles. I know that these are a lot of names for you to get used to, but it's useful to learn these, and it helps you remember the different things. So when you're studying this, write down these names, what keeps them together, what distinguishes them, what's a typical feature for all of them. So within this group, there are many, many photosynthetic organisms. And what is also interesting about this whole grouping, the chrome alveolata, is that there are many which are single-celled, but there are also some multicellular organisms in this grouping, and some of the largest organisms on Earth, in actual fact, which are the brown algae. And those are in this grouping, the chrome alveolata. So let's have a look. They, as I say, they all have these membrane-bound sacs, which are called the alveoli. Just completely unknown function, but it's just a morphological feature which they have. And here the groups are the groups in the true alveolates are the dinoflagellates, the apicomplexans, and the ciliates. You will have heard of some of those, I'm sure. So here, these are the alveoli. These are in this organism, they're associated with the flagella. And here they are at the sides, these kind of sacs at the side here. And again, their function, we don't know, but they're a very distinctive feature. In this particular one here, they're arranged here, but as we're going to see in things like the ciliates, they occur throughout the cell membrane, all over the cell. So our first group that we're going to talk about are the dinoflagellates. Dinoflagellate means two kinds of flagellum. And the dinoflagellates are predominantly photosynthetic. There are one or two which are completely heterotrophic, but they are predominantly photosynthetic organisms, aquatic, and they are found in freshwater and very, very abundantly and very diverse groups in marine systems, where they are extremely important elements of marine ecological systems. Much of the photosynthesis which takes place in the ocean takes place in the dinoflagellates. And they're, again, very diverse group. And one of the reasons some of you may know them, if you live close to the coast, periodically they will become so abundant, particularly in spring and in autumn. They may become so abundant in the water that they color the water. Often the dinoflagellates often have a red or brownish color, and they will stain the water this red color, which we refer to as a red tide. There is a problem with some dinoflagellates, however, and that is that some dinoflagellates produce very, very virulent toxins. And one of the things that can happen is that shellfish, mussels, clams, this sort of thing, can filter the dinoflagellates out as food, digest them, and accumulate the toxins and store them. And if we go then and eat those shellfish, we get a mega dose of the dinoflagellate toxin, which can be fatal. There's several distinct types of poisoning which occur because of dinoflagellate poisons. Here are some dinoflagellates. The dinoflagellate, the two kinds, refers to the fact that they have one flagellum which trails here and drives the cell through the water. They have another flagellum it's difficult to see, but you see this groove here that runs around the cell. Same here. Here it is here. There's a flagellum that runs inside that groove, and its responsibility is to turn the cell, because these are photosynthetic organisms. And so the cell gets turned like this in the water so that all the chloroplasts receive equal lighting while the cell is driven through the water by this flagellum here. These are very typical They have very diverse shapes, but these are just very typical. This is one called serratium. Here's a red tide. This is very typical. There's another interesting thing about the dinoflagellates, and that is many of them can produce light. They are bioluminescent. And when there is a red tide like this close to the shore, it can be very, very spectacular at night because the cells produce light when they are disturbed. So when the waves break, all the dinoflagellates produce this brilliant blue-green flash of light. It's very, very spectacular. And if you trail your hands through it, you can see streams of light. It's very beautiful. It's very, really worthwhile going to see if you have the chance to ever have the chance to do it. So this is a red tide, all dinoflagellates, which have formed a bloom in this gathered together here against the coast. Okay, completely and absolutely different to the dinoflagellates, but in the same group is this group here called the apicomplexans. And the apicomplexans are obligate parasites. Very, very strange group, but we know that they are related to the dinoflagellates for all sorts of reasons. First of all, structurally, they have alveoli, but also because we've studied their molecular biology and sequenced the DNA, and it is clear that the dinoflagellates and the apicomplexans are derived from a common ancestor. The most important of all is Plasmodium. And Plasmodium is the organism which causes malaria. Malaria remains probably the most important infectious disease of human beings worldwide, and it still kills millions of people a year. And it has been malaria throughout history. There's only one other disease which has ever rivaled malaria for the number of human beings that have been killed by it, and that is influenza. Malaria has been a major controlling factor of human populations, and it still is. And Plasmodium is a highly modified organism which infects, first of all, liver cells and then red blood corpuscles in the human being. And it is transmitted by the mosquito, and is transmitted by one genus of mosquitoes called Anopheles. And Anopheles is also infected by the Plasmodium. The Plasmodium passes through several of its life cycle stages in the mosquito, and then the rest of it is completed inside in human beings. And this apicomplexan, it gets its name from the fact that one end, at the top, those alveoli are modified. They're highly modified to enable the organism to penetrate cells. That's how it gets into cells. So this, just very briefly, is the life cycle of Plasmodium. Here's this part here in the mosquito, and in fact, most of the life cycle takes place in mosquito. That is where the sexual stages of the organism are present, and that is where meiosis takes place. So with meiosis, the organism produces these little cells called the sporozoites. And it is the sporozoites make their way to the salivary glands of the mosquito. When you're bitten by a mosquito, the mosquito injects saliva, which contains an anticoagulant, so it can drink blood easily. And that saliva contains sporozoites, which then enter your bloodstream. They make their way to the liver, and they breed inside the liver cells. This is one of the first stages. Inside the liver cell, they divide. These are all haploid cells because meiosis has taken place already. They are haploid. They produce these cells called merozoites, and the merozoites infect red blood cells. There can be several rounds of reproduction of merozoites, so you build up a very high population. And the parasite infects a red blood cell, actually gets inside the red blood cell and divides there, grows and divides. Eventually, some of the merozoites transform into cells which are adapted for sexual reproduction. And these are gametocytes. And there are male and female gametocytes. Those have to be taken up by a mosquito for the life cycle to be completed. Inside the mosquito, they produce egg and they produce sperm, which you fertilize, produce a zygote, and the zygote divides very rapidly by meiosis and produces more sporozoites. So that completes the life cycle. There's just one other thing to tell you about the mosquitoes. Mosquitoes are not, do not depend primarily on blood for their nutrition. In fact, mosquitoes are nectar drinkers, and they are very important pollinators. If anybody ever asks you, you're doing biology, tell me what mosquitoes are useful for, you can tell them. They're ecologically very, very important because they are important pollinators of plants. But a female mosquito, after she has mated with a male, she has and is ready to produce eggs. She requires a blood meal in order to produce her eggs. And after she has mated, she will go and seek a blood meal. She will take a blood meal, and if she's sufficiently fed, she may take several till she is fully fed. She will find a place to roost, and she will roost for several days until her eggs are mature, and then she will fly out, and she'll find a water body, and she lays her eggs in water. And the initial stages of the mosquito life cycle are aquatic. That is why one of the most important ways in which to manage a mosquito population, to reduce mosquito populations, is to reduce the number of places which have got stagnant water in them where mosquitoes can breed. Okay, so those are the AP complexes, very, very, very important in terms of human health. There are also, I should just tell you, almost every vertebrate group has their own AP complexes that infect them. Birds have their own malarial parasites. Even lizards, I believe, have their own. So many different vertebrates have an association with the AP complexa. Okay, here's another group in the alveolata. This is another very, very important group, single-celled organisms called the ciliates. And the ciliates are so named because they are covered with a forest of little tiny, they like exactly the same structure as flagella, excepting that they are very short. And these are the cilia that it derives its name from. The whole outside of the cell is covered with these cilia. There is a rigid outer covering called the pellicle, and all of the cilia beat in a coordinated fashion, first to move the cell through the medium, but second of all, to conduct a current down here into this groove here called the oral groove. And at the end of the oral groove, food vacuoles are made so that the ciliate beats food particles into the gullet and ingests them by forming a vacuole here. The ciliates have another characteristic which unites them all, and that is that they have two nuclei. They have one large nucleus and then a micronucleus, and their reproduction, the cell division, is extremely complex, and they can, in general, they reproduce asexually. They simply grow and divide, grow and divide, grow and divide, producing identical copies over and over again, but they can reproduce sexually as well. They actually get together and conjugate, and they exchange genetic material and in a very complex fashion to accommodate the fact that they have two nuclei. So here's one under the microscope. This is very, very typical, granular looking sort of cytoplasm. But you see this, it looks like petals of a flower in there. This is one of two contractile vacuoles which they possess. Here it is here. There are canals which feed into the central vacuole, and they are pumping water in. This is a way to resist osmotic pressure. Water is flooding, if this is freshwater ciliate, water is flooding into the cytoplasm continuously because the inside of the cytoplasm is more concentrated than the outside of the cytoplasm. The outside is more dilute. So water continually pours into the cell through osmosis. And to resist that, the cell continuously pumps out water. The ciliates have two contractile vacuoles. There's one there, and there's one there. And if you look under the microscope, you'll see one filling just as one empties, and they beat like that. You see one fill just as one's emptying, empty fill, empty fill. So it's a coordinated pumping of water. Very, very big group, very, very diverse. There are many, many, many ciliates in all sorts of environments, in the soil, in water. When I say in soil, I mean in the water of soil, in the liquid of soil, in fresh water and in marine systems. And also very often in the gut of different organisms, where they perform many different kinds of function. In the termites, for example, termites feed on wood, which is mostly cellulose, which is very, very difficult to digest. And termites maintain colonies of ciliates, which actually are responsible for digesting the cellulose together with prokaryotes, which the ciliates have an association with. The same kind of system exists in animals. For example, in ruminants, in cows, and even in things, in many, many different herbivores, there are large colonies of ciliates in the gut, which are commensal. They are there to digest cellulose and provide the products of that digestion to the organism that hosts them. Okay, so that's these groups here, the dinoflagellates, apicomplexans, and the ciliates. This other group here, these all of these diatoms and the brown algae here, and we're not going to talk about the golden algae. These here are grouped together because they share a distinctive kind of pigmentation. And both of these, if you look at them, the diatoms and the brown algae, they have a distinct golden brown look to them because of the particular pigments that they have. So let's just have a look. First of all, this group, they're called the straminophiles, and they are all united by yet another morphological feature, and that is each of these, if they have a flagellum, not all of them have, the diatoms don't have a flagellum, but if they have a flagellum, for example, in the brown algae, the sperm have a flagellum. If they have flagellum, then at least one flagellum has a particular structure. It looks like a brush. They usually have one smooth, and then they have one hairy flagellum that looks like a brush. I'll show you a picture in a minute. And the only two groups that we're interested in for the straminophiles are the diatoms and the brown algae. Both very, very, very important groups, particularly in marine systems, although diatoms occur in fresh water as well. In any case, here's a Straminapile flagellum here, and here you can see the hairy, it's called the hairy flagellum, and it looks like a brush there. And here's the smooth flagellum. All the Straminapiles, when they have a flagellum, when they have at least one, which is hairy. So that's what unites them. The diatoms are a wonderful group. They are single-celled organisms. They are photosynthetic, and they build these absolutely beautiful cases around the cell. It's called the test, T-E-S-T, test. And the diatoms, the test consists of two halves, and the two halves fit together like a petri dish would, like that. Here you can see, here's the top here, there, and here's the bottom half of it here. And they can be extremely, extremely elaborate, beautiful to see under the microscope. As I say, they occur in freshwater and in marine systems. Very interesting, very diverse group. And in marine systems, they can be extremely abundant. And so abundant, in actual fact, that the tests, when the cells die, the tests are made of silica, silicon oxide. It's glass. That's, in fact, most glass is derived ultimately from diatom tests, as I'll tell you in a minute. But they can be so abundant that when the cells die, the tests, which are heavier than water, fall out of the water column, and they accumulate in deposits, which in some parts of the ocean are thousands of feet thick and have been accumulated over millennia. Those deposits can also become consolidated and can form what is referred to as diatomaceous earth. Those of you with swimming pools may have seen that term. Diatomaceous earth is used for many different things, but one of the things it's used for very often is as a filtration mechanism. But it's also used as a source of silica for glass and for other things as well. So here are some, these are all marine diatoms. You can see hugely diverse in their shape, all these different species, genera and species with their different shapes. They are photosynthetic. They have a distinctive golden brown color to them. And those, the deposits of the tests, first of all, in marine systems, it's called diatomaceous ooze. And an ooze is a mud, essentially, which is predominantly the diatom tests. But that can become consolidated so heavily that it can begin to form rock. And under certain circumstances, the silica from those diatomaceous tests can form actual rock, a glassy kind of rock, which is a chert and often called a flint. And here is one here, and it's glassy because it's silicon oxide, and very, very frequently was used to make stone tools like hand axes, arrowheads, and that sort of thing. Very often in Stone Age times, or actually not in the Stone Age times, right up to modern times. Native Americans, for example, frequently used cherts and flints as a basis for making stone tools, arrowheads, and that sort of thing. So here's a raw flint, and here's a hand axe made from. Okay, so the other group of Straminopiles, which are very important in marine systems, are the brown algae. And some of the brown algae are absolutely enormous, as I say, amongst the biggest organisms in the world. They are just incredibly, incredibly long bodies to the plants. I call them plants, but they're not actually a true plant. It's an alga. And the brown algae usually consists of the following parts. They have a holdfast, which ties them to the rocks that they grow on. Then they have a long, the equivalent of a stem. It's not a true stem, called the stipe. And the stipe, it may extend very, very long. It may be hundreds of feet long in certain species of brown alga. Then somewhere on the stipe, they have flattened equivalent of leaves. They're not true leaves, called the blades, which are the major photosynthetic. The whole organism can photosynthesize, but this, with all of its surface area, the blades are the major photosynthetic organ of the brown algae. A very ancient group, by the way. This is the brown algae have existed for a very, very long time. They exhibit what is referred to as an alternation of generations. That is, they move between haploid N and a diploid generation. And the haploid generation is usually rather obscure. The diploid generation is the major plant that we see. And that is also called the sporophyte. So here is the sporophyte here. And this would be a typical brown alga or a kelp that you would see on the west coast, for example, of the United States, where there are many different species. And here is the sporophyte here. And the sporophyte has modified reproductive cells called the sporangia. And the sporangia undergo meiosis, and they produce spores, which are N. And there is some of the spores are the equivalent of female, like an egg. Some of them are the equivalent of male, or N, or male plants, really. And they produce these little tiny obscure filamentous plants. Here's a female plant here, the equivalent of a female. Here's the equivalent of a male plant. And these little plants here produce egg, and they produce sperm. So you've got male spore, female spore, producing female plants, male plants. And the male plants produce sperm. The female plants produce eggs, which unite in fertilization. This is the N generation. This is the haploid generation here with the separate male and female plants. So that's the haploid generation there, unite to form diploid. And diploid goes on to form a new sporophyte, which is the major plant that we see. Okay. I just need to flip back. I want to flip back all the way here. Oh, sorry. I thought I had it on there. Okay, we're going to move on now to our next group, which are called the Rhizaria. And here they are here. The two groups that we're going to talk about are the foraminifera and the radiolarians here. Now, both of these, we won't talk about these. Both the foraminifera and the radiolarians are essentially like an amoeba, a single-celled amoeboid form, which have around them an elaborate shell that they make. The shell is quite different in the foraminifera and in the radiolaria. In the foraminifera, it's made of calcium carbonate. In the radiolaria, it is made of silica, again, silicon oxide, as was in the diatoms. So these groups, there's nothing much, I have to tell you, that obviously unites them. And some people were really, really objected to the foraminifera and the radiolaria being joined together because they regarded them as being so very different to one another. But DNA evidence tells us that in fact they are closely related and belong in a single clade. So they, just like other amoeba, they can move, they produce pseudopodia. And the pseudopodia, they're first of all each of the radiolaria and the foraminifera, they've got a rigid test, you'll see it in a minute, but it has pores in it, and the cell can extend these pseudopodia, which means false foot, through these pores for locomotion and for feeding as well. Foraminifera and radiolaria actually are predominantly float. They are planktonic and they float in the water. And both of those groups are predominantly marine. They are marine groups, not freshwater. So let's have a look. Both groups, foraminifera and radiolaria, can be extremely abundant in marine systems. And again, when they die, those tests can rain out of the water column, fall to the ocean floor, and form oozes, which can ultimately be turned into rock. So this is a foraminifera. It's a single cell, but it's got these chambers to its cell, like this. You can see one here. And the whole thing is perforated by pores. You can see there, all these little tiny pores all over the place. And it extends pseudopodia out from those into the water. The pseudopodia do two things. The first thing is that they actually catch food, and it is brought down to the cell body to be ingested. The second thing is this actually enables them to float. It stops them from falling out of the water column very rapidly. It enables them to float much better. So those are the foraminifera here. There are foraminifera all over. There are some sedimentary ones, planktonic ones. You find them in many, many different marine habitats, many, many different types like this. And foraminifera are found in all marine deposits, whether they are fossilized or modern. And the foraminifera shells are very important markers of oceanic climate. Certain kinds of forams only occur under specific conditions. So they are a very good marker of previous oceanic conditions. If you look through the sediments, isolate the foraminifera shells and identify them, it gives you a record of what conditions were like when that sediment was created. Very, very diverse in their shapes. When they become absolutely compacted into rock, this is what they form. It's chalk, the same as, well, we seldom see it these days, the same as blackboard chalk. That is, those are compressed foraminifera shells and calcium carbonate. That is exactly what they are. And here you can see, look at the depth of these deposits here, and you'll realize just how abundant forams actually are in marine systems. So these are white cliffs in England, the White Cliffs of Dover. In fact, I'll tell you that if you go to the south of England, practically half of England, almost all of the south of England is actually chalk. Much of it is chalk. The geology is a chalk substrate. So this is the other group of Rosaria. These are the radiolarians, also an amoeboid form. They make these elaborate and very beautiful glass shells or tests. Again, you can see they are perforated with pores, out of which the pseudopodia are extended, just as they were in the foraminifera. Again, these are marine organisms, and they can be so abundant that they will actually form radiolarian oozes in the sediment. predominantly radiolaria or predominantly planktonic. Foraminifera, you do get many sedimentary ones as well, as well as planktonic ones, but radiolaria are predominantly planktonic organisms. You can see the elaborate tests. These would all be different genera and species of radiolaria. Okay, so here's our next group. Remember, these were actually placed together with the Rosaria. Here, we believe that there's some relationship between them, although the relationship is not exactly clear. DNA suggests to us that it is. But the archaeplastids are a monophyletic group. That is, they have a clear common ancestor. And that ancestry is very significant because we believe that it is in this group that the endosymbiosis of the chloroplast first occurred. It is in something like an ancient archaeplastid, was the first cell to take in a cyanobacterium as an endosymbiont and adapt it to allow it to become a chloroplast. And there are two really huge groups which have been derived from this kind of process. Now, in your textbook, I think if you go back to, I think, the history of life, there is a lot of information there about the different kinds of endosymbiosis that took place to produce our modern eukaryotes. But here in the archaeplastids, we see two which are truly significant. The first is this group here called the red algae. Now, the red algae are not a dominant group. They are not a huge group. They're quite diverse, but they're definitely not one of the dominant algal groups. But they are a very, very, very ancient group. And we find them actually, we find fossilized red algae in Ediacaran fauna, more than 1 billion years old. So the red algae are an ancient group, and they may well have been one of the first multicellular groups as well. In addition, they established a very distinctive endosymbiosis. Their chloroplasts differ from all other chloroplasts in several aspects. That's why they are red. It gives them, their pigmentation gives them their red coloring, although they have chlorophyll and everything else as well. So they're a very ancient, very interesting group indeed. And we have a lot to learn from the red algae about how the eukaryotic cell arose and how it moved from being a single cell to being multicellular. The red algae, in addition, have very, very complex life histories, which we won't discuss at all. But they can have very extremely complex life histories with an alternation of generations. They're a fascinating group. But they are a kind of evolutionary dead end, if you like. There's not, when we talk about the other groups here, you will realize that their evolutionary development of the red algae was limited. In this group here, the chlorophytes. Now, the chlorophytes are also colloquially referred to as algae, or the green algae. We believe that the very, very early on, the green algae, a far ancestral green alga cell, also underwent an endosymbiosis with a cyanobacterium to acquire a green chloroplast. And that chloroplast was in fact identical to became identical to the plant chloroplast. And it is in this group the chlorophytes that plant photosynthesis developed, that the plant's eukaryote cell with a green chloroplast undergoing photosynthesis. It was in this group, the chlorophytes or green algae, that occurred. So first of all, there are many chlorophytes. They're freshwater and marine. They're very diverse. They have all sorts of different structures to the body of the organism itself. But there is one group of chlorophytes which we are particularly interested in. And they are these. They're called the charophytes. So first of all, let's just have a look. Here, these are some red algae for you. They vary a lot in a lot of them are these kind of these kind of elaborate sort of branched filamentous algae, and but some of them are flat. And these ones you know well because this is Porphyra, and this one here is Palmaria, but there is another one widely grown in Japan called Palmaria. I've called Porphyra, which is the red alga. It's just a flat blade. It's only 2 cells thick, the blade, and that is what we use for wrapping sushi. And red algae are commonly eaten all over the world, very often as a salad, and then of sushis, they're the sushi wrapping. So they're a very important food item, the red algae. So the green algae, are they named for the fact that they are this bright green, grass green color. And that is from the chlorophylls that are present in them. These two groups, the chlorophytes and the charophytes. Here, this is a typical chlorophyte. This is Volvox, which is a freshwater organism, although you will see they're not called Volvox, but you'll see some of the ones in marine systems. But there are many multicellular chlorophytes as well. Here are some here. This is very common. You'll see this everywhere on the edges of the sea, on rocks and everything. This is alva or sea lettuce. It also sometimes eaten. Here's a more elaborate one. This is called lerpa. They're very, very diverse in marine systems and mostly intertidal. That is growing on the edges because that is where they get sufficient light to be able to photosynthesize. Very important ecologically in marine systems and in freshwater systems. But for a biologist, they are fascinating because of this particular group within the whole group altogether. And this is this group called the Charales. And Chara, this is typical. There's another one called Nigella. This is very, very plant-like. It's multicellular. But what is important about it is that it's multicellular in a particular way. It's not just a collection of very similar cells. The cells actually are starting to differentiate. They're starting to differentiate into things which we look at and we can recognize as being precursors to features that we see in the plants. And for that reason, sorry, let me go back here. If you look here, you'll see here are the charophytes. They are joined, in actual fact, to the land plants. Now, the land plants are excluded here. They are not protists at all, okay? They are not protists, but the plants are closely related to the charophytes, and we believe that this is a valid grouping. The chlorophytes, the charophytes, the land plants. This is a valid grouping which reflects their evolutionary history. That the land plants, in fact, are derived from the land plants, charophytes, and chlorophytes are derived from a common ancestor, an ancestral archaeplastid that had a green chloroplast as an endosymbiont. Okay, so that is the reason that we pay particular attention here to the caryophytes. We'll hear about it again when we talk about plant evolution and plant biology later on in the semester. Let me just see where I am. Okay, so I'm just going to leave this for the moment, and I'll begin our next class by talking about the Uniconta, which is another group which tells us a lot about evolution of the eukaryote. So for the time being, that's it for today. Thank you.